This video looks at regulation of the cell cycle and is part of cells as the basis of life topic. Now, of course, the cell cycle is a really important thing for all cells to complete if they are to divide and make new cells, which is required for all growth of multicellular organisms. But it's also really important to be regulated because it's such an investment of resources to go through the cell cycle and make a whole new cell and divide. So, as you would think, this has to be a regulated process so that cells don't commit to the cell cycle unless they are ready to do so. So one way of looking at the cell cycle would be like this. As we've seen before, there's a large phase that we call interphase going around here that requires growth phase 1, sometimes called gap 1 as well, DNA synthesis where the DNA is replicated, and growth phase 2 before we undergo cell division. In this case, we're looking at mitotic cell division, which involves mitosis, and then you have cytokinesis at the end. There are three major checkpoints around this process that the cell needs to go through in order to proceed through the cycle. The G1 or S checkpoint, G2 checkpoint, and the M checkpoint. And the reason why, again, is because there's a major commitment of resources that is required at each of these, and the cell wants to check that it's done everything right before it moves on. Clearly, for instance, here, the checkpoint here is before we go into the process of copying all the DNA. Millions of base pairs, lots of nucleic acids required, lots of energy used. The synthesis process is a major undertaking. The cell needs to check that it's ready to do so and that it wants to do so. So it needs to check, are we going to commit to going through the cell cycle and making a whole new cell before I start making all of this DNA? And do I have the resources to do so? So the S checkpoint, G1 checkpoint, is all about checking to make sure that the cell is ready and committed to going through this. Because, of course, once you've copied all that DNA, that cell really needs to go through and complete and become two new cells. Here is checking that everything was done properly. So if the replication of DNA wasn't correct and that we're not prepared for the next process of cell division, we might need to wait or pause at this point and make sure everything's ready before going on, because if the DNA hasn't been copied properly, again, there's no point in going through mitosis. This final checkpoint here is just making sure that everything's happening and proper before cytokinesis happens. So we have checkpoints around here that need to be passed in order for the cell to move through the cell cycle. Now, completion of these checkpoints relies on both internal and external factors, but they interrelate to each other. By internal factors, what I'm saying is the cell is checking its own state to make sure that it's ready to go through each of these and that the DNA has been copied, or that it's got enough energy, or that the proteins required have been manufactured. And it keeps track of this through some internal proteins. But also the external state, meaning in a multicellular organism, there are hormones and other physical constraints that are regulating whether a cell should and can divide. So, for example, in terms of the physical state, cells in close contact with each other will undergo a process called contact inhibition, meaning that if the cells are already crowded in that area, there's no need for the cells to keep growing and dividing. So in a wound situation, there's no contact on one side of the cell. It knows that there's a gap that needs to be filled. It will go through cell cycle and grow and fill that gap until it meets the cells on the other side. Contact inhibition kicks in and the cells stop growing to heal that wound. Also, obviously, in terms of development, we have hormones that are signaling cells to grow or to stop growing. So that's some of the external signaling, physical from the contact and chemical from the signaling. And that combines with the internal signals to regulate and process. So some of the internal signals are regulated through these proteins here, MFP, MPF, CDK, and cyclin. So MPF, mitosis promoting factor, is actually made up of the other two proteins. So if we just focus on the right hand side, to pass through this checkpoint and know that mitosis is ready to happen, we actually need this mitosis promoting factor, which is a combination of CDK and cyclin. The two proteins lock together to make a combination protein here, and that one is the one that signals that we're ready to go through this checkpoint at G2 and mitosis can go forward. So actually what happens is that CDK is always present in the cell and made at a fairly regular level. And so what's important here is the level of cyclin that needs to be available to combine with CDK. And so what we see is that at the beginning of the cycle, G1, cyclin levels are very low, and they build up in the cell as it grows and goes through those other phases, till it's enough cyclin there that it can combine with the CDK to make MPF. And so suddenly we have a quick rise in the amount of MPF. That signals that mitosis can happen, and then as mitosis is happening, the cyclin is degraded, drops down rapidly, and therefore MPF is degraded as well, and we start back in this situation where we need to start making more and more cyclin to build up. So cyclin is basically like a clock, or the sand in an hourglass. As it's building up, we're seeing the progress of time in a cell. So as it builds up, it's a signal that the cell has grown and it's produced enough protein. This is a kind of like a marker protein, and hopefully by showing that this protein is growing, it knows that the rest of the cell is growing as well. So it's one of the ways that the cell can keep track of its growth without having an actual clock, obviously, or any way of measuring its own size. So it knows it's been growing for a while once the cyclin level has increased to a sufficient point where MPF can form and mitosis is triggered. Other things then that can control the rate of cyclin production, or even CDK production, and other factors within the cell would be external signals. So the MPF, cyclin, and CDK are internal things, but they're controlled, and the amount of each of them can be controlled through hormone signaling. So the way that works is that hormones and growth factors in the external environment carried by the blood will bind to receptors on the plasma membrane, and these will cause a signal transduction pathway. These relay proteins will actually then change gene expression. So genes will be turned on and turned off. So for example, if you increase the production of cyclin, you will hasten the cell cycle. And so by changing protein expression, you can adapt or make changes to the cell cycle control system. One other really important, slightly separate but important part of this is when the regulation doesn't work properly. So clearly, as I said before, cell cycle needs to be regulated so you can turn on and turn off cell growth. What happens if the regulation goes wrong? Well, if you've turned off cell growth, then the cell sits there and doesn't really do much. But if you've accidentally turned on cell growth, cells can continue to grow unchecked and uncontrolled. And this is basically what cancer is all about. So you have normal cells that should grow and divide, but if they grow in an uncontrolled way, you start to see the beginnings of a tumor. So how might this happen? Well, carcinogens, cancer-causing agents, are one of the things that we know causes cancer. These are chemicals and other agents that can cause DNA damage. Now, normally, if you're damaging DNA, it's not going to affect cell cycle, but just remember, if we go back to this slide, that a lot of the genes that control cell cycle are just regular genes, and if you somehow damage that or change its function, you can actually affect cell cycle. So cancer happens when one of the cell cycle control genes is damaged or upregulated, causing increased cell growth. So once this has happened, the regulation ceases to occur and the cells can continue to grow. This is a primary tumor. If those cells become invasive and go through the blood system, they can cause secondary tumors in other parts of the body, which of course is extremely damaging. So cancer is basically a disruption of the regulation of cell cycle, causing cells to grow in an uncontrolled way.